Hello, everyone. Thank you for joining us today, and thank you to LabRoots for this opportunity to present some of the work that we've been doing at NIST over the past few years regarding the NISTmAb and biopharmaceutical reference materials. So I'm John Schiel. I'm the project coordinator for the NISTmAb reference material. I've been with NIST for about the last 10 years working on biopharmaceutical characterization measurements. And again, thank you very much, and looking forward to the opportunity to share some of our work. So first of all, we have to start out with a NIST disclaimer. We will be talking about publicly available reference materials today, and so I will be showing some values on the screen. But we do urge stakeholders to please check the most recent certificate for the reference material if you are interested to get any of the most up-to-date measurements and up-to-date values associated with the individual reference material. So I wanted to start by giving a little bit of a background about the National Institute of Standards and Technology. We are actually a non-regulatory agency within the Department of Commerce. So initially, it may make you wonder, why is there a science-based organization in the Department of Commerce? And the answer to that is we really are responsible for the physical test standards, measurements, and calibrations for a whole variety of individual measurements in individual industries throughout the United States. So we have a very unique mission to promote innovation, industrial competitiveness by advancing the measurement science, the standards, and the technology with a specific focus to enhance the economic security within our nation. So I have a couple of pictures of reference materials here. The first on the bottom left here is actually a picture of a cholesterol in human serum. So one example of how NIST actually contributes to various industries in this particular case is you may go to the doctor to get your cholesterol checked. And we have a reference material that diagnostic companies can actually use to calibrate their measurements, calibrate their instrumentation. And we provide a material that's actually traceable to the actual SI unit of the kilogram. So we know exactly how much cholesterol is in that vial, and everyone throughout the United States can then calibrate their individual measurements on this particular vial of cholesterol. So we actually do this for a wide variety of individual technology and measurement types. My particular expertise is in the biological measurement space, but again, NIST has programs in every type of measurement science that you can imagine. This goes from advanced mathematics and statistics to computer science to cybersecurity, and there's even a fire research program at NIST. So we have a very broad range of expertise for all scientific and engineering backgrounds. Our core strength is actually in biological measurements, and this is primarily harnessed in two individual divisions, the Biomolecular Measurement Division and the Biosystems and Biomaterials Division. So, of course, a lot of the biomeasurements kind of cross paths between both of the individual divisions, but overall we have a very broad range of types of analyses that we can do. So in the upper left-hand corner, we can talk about measurements for individual biomolecules. So this can be things like DNA, RNA, proteins, and metabolites. We also can measure the analytics associated with biomanufacturing. So in the case of biopharmaceuticals, we may use cell culture, for example, to produce proteins or RNA, DNA, or portions of protein therapeutics, viral vectors, things like that, that may actually be used as a therapeutic modality. So we also have a program in cell culture and analysis of the actual biomanufacturing process. On the right-hand side of the circle, we have quantitative tools for cell characterization. This could be things like cell identity, morphology, viability, really looking at the individual cell characteristics, but also in the bottom right-hand corner, models and understanding cells and population and their behavior as well. So we really do have an infrastructure to measure the entirety of biological systems from cells to the individual molecules. So with that measurement system, we've actually had a program specifically in biomanufacturing. So now we're kind of getting into the protein therapeutic space, which is where we spent approximately the past 6 or 7 years. Again, we have a very wide program, but this is focusing mainly on the monoclonal antibody type characterization and analysis that we've done over the past few years. So getting a little bit deeper into the individual types of measurements we're able to do. On the left-hand side, we have protein stability and biophysics. So I'll mention one of our reference materials that's available that I won't talk about too much today, which is SRM 1989. So this is actually a surrogate protein particle material. It's made out of a polymer that has refractive index characteristics very similar to a protein particle. So you can actually see some image of these protein particle standards in the bottom left-hand corner. This is actually what very large aggregated proteins look like under a microflow imaging. Very similar characteristics, and this standard is actually between 0.5 to 25 microns in size. and it can be used to calibrate various types of instrumentation, such as microflow imaging for those particulates. We've also got some very unique capabilities within NIST that are rather uncommon throughout the United States, including neutron scattering and small angle X-ray scattering. These techniques can be very useful in looking at the dynamics of proteins, which is sort of an emerging measurement area in the field of biopharmaceuticals. Moving down to the bottom of this triad, we have our production cell science. So obviously biopharmaceuticals like monoclonals are prepared in various cell systems. This can be humanized cell lines, murine cell lines, etc. So we are actually developing some reference cell lines that will express a NISTmAb non-originator molecule that I'll talk about a little bit later in the presentation. But we also have proteomics-based characterization for things like the actual CHO and other cell lines that are producing those materials. We're getting into proteomics of CAR-T, another therapeutic area, and really looking at essentially the production cell line characteristics and how we can define things like proteomics, metabolome, and genetic diversity of those production cell lines. The upper right-hand corner is where my lab spends the majority of our time. My lab actually looks at primary structure and quality of therapeutic proteins and other therapeutic modalities. So we predominantly use mass spectrometry, capillary electrophoresis, and HPLC to characterize things such as the primary structure, glycosylation, and other protein post-translational modifications. Within NIST, we also have a very excellent program in higher order structure, ranging from nuclear magnetic resonance, HDX-based mass spectrometry, and recently we've added electron microscopy capabilities. So we really do have an emerging infrastructure for higher order structure. And of course, looking at things like biological activity through surface plasmon resonance, secondary and tertiary structure measurements as well. So as you can see, NIST really does have a very broad range of measurement capabilities. This is absolutely not an exhaustive list, but certainly we are able to measure the protein characteristics of relevance to the quality and manufacturing of these products. So when I talk about measurements on biopharmaceuticals, obviously there's a wide variety of analytical measurements. But when we start thinking about a monoclonal antibody, we start thinking about what attributes about this protein we actually want to measure. And if you look at a monoclonal antibody, initially you start thinking a reference material that's a common material that can be shared among people to do things like harmonize analytical methods. It's hard to choose which monoclonal antibody is it that you'd actually like to use for that analysis. When we start to look at the molecule, every monoclonal antibody has 2 light chains, it has 2 heavy chains. You can see the individual cis markings on this particular structure. Those are disulfide bonds. Every monoclonal antibody also has these disulfide bonds, either within the same chain, such as the cysteine 22 in the upper left-hand corner, or between the 2 chains, as you can see in the cis 222, 229 in the middle of this figure. So in addition to that, you can see we have these sort of different shapes. There's the triangles and the circles and the squares that are sort of peppered throughout this individual molecule on both the heavy and the light chain. So these are meant to represent various low abundance post-translational modifications. So on the right-hand side, I have a couple of different post-translational modifications. For example, methionine oxidation, where the sulfur residue of this individual amino acid can get oxidized. We have asparagine deamidation, where the NH2 group can actually be changed to an OH group. So we're going from an asparagine to an aspartic acid. And then very importantly, we have glycosylation. So in the bottom right-hand corner, you can see this polypeptide chain with the asparagine highlighted. And I have 3 different glycans highlighted here. Now I can say that this individual glycosylation site, which there are 2 of on the NISTmAb material, 1 on each heavy chain, there's actually been 40 different glycans that we've identified on this material. And any individual asparagine at this particular location on any individual molecule of the NISTmAb can have any one of these 40 glycans attached. So you can imagine that there's significant heterogeneity added to the structure or the population of these monoclonal antibody materials simply by this one post-translational modification. So going back to the structure on the left-hand side, thinking about the monoclonal antibody as a platform, it's true that each individual type of monoclonal antibody is going to have different levels of methionine oxidation. They'll have slightly different post-translational modifications. They'll even have slightly different sequences at the top of the structure in the FAB region. But when we start thinking about these attributes and how we're going to measure them, every monoclonal antibody we're going to approach in the same way. We're going to use the same measurement techniques to look at these attributes, whether it be the primary sequence, post-translational modification, aggregation, size variance, etc., we're going to approach that measurement challenge in the same way. So when you start thinking about it in that direction, it really is possible to have a common material that can be utilized to do some harmonization throughout the industry. So in this case, we started to talk to our small biotech, our large biotech, and our federal collaborators, and we did actually get that message that everyone should be using a common standard. It would be great to have something that was shared, that we could have joint system suitability criteria. And in the bottom here with large biopharma, the more robust these standards are, the better off we'll be when we start to develop attribute-specific monitoring assays, which I'll talk about a little bit later today, that can really look at each of these individual decorations on the monoclonal antibody material in very high specificity. So in comes the NISTmAb reference material 8671. So the NISTmAb is a publicly available and biopharmaceutical grade monoclonal antibody. So this is publicly available from NIST. We actually sell our materials on a cost recovery basis. So this is not for profit. So we try to make these as low price as possible to try to get the greatest impact and the most distribution and availability of these materials. They're also available for certain collaborations within NIST, and I'll talk about some of that differentiation with the life cycle plan. But essentially, this material is publicly available. It's intended to be voluntary, meaning that manufacturers of biopharmaceutical products are not required to use this material, but we hope that they use it as sort of a product-neutral standard that they can use for the internal and external collaborations, making any data that's generated about this molecule pre-competitive that the entire community can utilize to advance the field further. The material has been very exhaustively characterized. In fact, we started this project in 2014 and 2015. In the upper right-hand corner, you can see this ACS Symposium series. This is actually a book that we had recruited about 100 individuals throughout industry and academia to apply each of the analytical methods on the bottom right-hand corner. Each of the individual chapters talks about a certain characterization analytical technique. So it actually has sort of a background and a how-to manual as the introduction, and then it has real data, discussion, materials, and methods on characterization of the NISTmAb. So this is sort of a documentary historical standard as well as on best practices for each of the individual analytical methods. We then moved forward from that exhaustive characterization, and we did a series of analytical qualifications. So essentially evaluating the precision, robustness, and quality of each of those analytical assays that could inform on the stability or purity of the molecule. This is highlighted in the analytical and bioanalytical chemistry cover on the right-hand side. I'll talk a little bit more about these articles, but herein we describe the reference values, the stability and the homogeneity of this material that we've assigned at NIST to assure the long-term availability of this material to our stakeholders. So in the end, the NISTmAb material really does provide an industry-wide shared resource. It can be utilized for novel technology development, for de-risking new analytical approaches before they're applied in a regulatory filing. And we think that this can be used really for a variety of collaborative studies to sort of address shared challenges within the community, to really assess current analytical capabilities and state of the art, and again, evaluate the sort of regulatory readiness of new and emerging analytical techniques. So we've been focused since release of this material in 2016, over the past few years then ramping up our analytical efforts to develop innovative technology and also again perform interlaboratory studies to evaluate the current state of characterization of some of these new techniques. So before I get into talking about the qualification analytical methods and sort of the interlaboratory studies, I wanted to mention something about the life cycle management of this product because it really is unique for a NIST reference material. And although it's modeled after a biopharmaceutical development program, it really is unique in the fact that we need to make sure that this material is available with the same attributes for the long term. So in the upper left-hand corner, you can see the bulk material that we received. This is the bulk NISTmAb material, 100 mg per mil. We actually diluted one lot of this material to make it as our primary sample. So this is our in-house primary standard material and also our collaboration material. We use this for the ACS books that I mentioned before, as well as our early method qualification. We then took the remainder of this bulk material And as you'll see in the top side here, we've actually homogenized multiple lots of the bulk into what we're calling the homogenized bulk. So this is to ensure that we don't have a lot-to-lot reproducibility issues down the road. So we actually homogenized a large quantity right up front, froze it at -80 degrees Celsius, and we have multiple 1 liter aliquots of this material as our homogenized bulk. Now, each container of homogenized bulk can be diluted 10-fold, and we can make 10,000 units for each individual dilution lot, where we've initially prepared 3 of these dilution lots, 14HB-D-001, for example. Each lot, again, 10 mg per mil, 800 microliters per vial, that are now available to the stakeholder community. So in the bottom right-hand corner, this is sort of also our measurement hierarchy. So you can, of course, see the working reference homogenizing these bulk materials in the middle into the 14HB. That 14HB goes into the top near the commercialization letter. And here's where we have our individual dilution lots that are available to the public. In the middle, then below that, you'll see the NIST working reference lot. This is actually the same material. It's our first commercial lot, but we use this internally for all of our analytical development, our routine research, et cetera. So we're sort of doing a continuous monitoring of this sample as time goes on. And then you'll see that primary NIST primary reference lot in the bottom. This is our sample that on an annual basis, we do a direct comparison between anything that's our internal primary standard as well as whichever lot is currently available to the public. And we do this correct direct evaluation to ensure quality and consistency of this material as it goes to the public. So when I mentioned quality and consistency, this is a highlight of those analytical and bioanalytical chemistry papers. These were actually all published online, and they are the analytical methods we're using to monitor the quality of the NISTmAb material. So in the upper quadrant, we see this first paper really discusses the life cycle and the quality plan in more detail if you're interested in that. We follow this by 3 individual papers, one on size, CIAF and CZE, charge, CIEF and CZE. The middle, we have a qualification of methods based on size. So this is capillary electrophoresis again, using sodium dodecyl sulfate, size exclusion chromatography, dynamic light scattering, and microflow imaging. And then we have a paper on the identity using peptide map to confirm the identity of the NISTmAb. And finally, we have the paper on the value assignment, stability, and homogeneity, where we've actually assigned values to the lots available to the public. All of this final paper, the value assignment, stability, and homogeneity, is summarized very succinctly in the report of investigation that actually comes to the individual user. And this is where you can find those values associated with the material to know what our measurements actually revealed for the individual attributes of charge heterogeneity, size heterogeneity, and identity of this individual material. So once we've had this material now publicly available, we now have initiated interlaboratory measurement comparisons. So this is actually an extension also on the ACS books, where we've selected a number of emerging analytical techniques, and those that could sort of use an interlaboratory assessment of things like the potential sources of variability and precision amongst these individual assays, how these assays are actually being performed in the biopharmaceutical industry. So it could potentially identify places where additional development may be needed. It also informs NIST on potential reference materials, and I think most importantly, it fosters collaboration amongst the global biopharmaceutical community to really advance these analytical technologies to the next stage. So most of these have been published. The 2-dimensional NMR, glycoanalysis, and HDX round robin have been published, and I'll share just a little bit of information about the multi-attribute method in that interlaboratory study, which we've recently submitted to MABS and should be available in the next couple months. So first and foremost, what is the multi-attribute method? This is something you may have heard about in the literature at various conferences, and it really is an extension of what we think about as peptide mapping. So in the upper left-hand corner, we have our monoclonal antibody. This can be digested using an enzyme such as trypsin, and you can see then these gray bars indicating the peptides, and then the black bars indicating the glycopeptides that may be making up that individual monoclonal antibody. We'll then analyze these peptides and glycopeptides with liquid chromatography hyphenated to mass spectrometry. And we can use then the mass spectrometry to, again, confirm that primary sequence, identify and quantify post-translational modifications, and perform site-specific glycan identification and quantification as well. So the multi-attribute method actually starts during the development phase, much as a mass spectrometry-based peptide mapping would, whereas each individual peptide or group of peptides coming out of the chromatograph. In this case, in the MS1 box, we're actually showing a fictional example where we have 3 peptides coming out at the same time. The mass spectrometer will select each of those individual peptides. Moving from left to right across the collision cell will actually collide each peptide with a collision gas. The peptide will fall apart into its constituent components, and we can essentially then piece back together this puzzle from the MS2 fragmented spectra back to tell us the identity of the individual peptide that was measured in the MS1, and then we can combine all of that information to essentially confirm the primary structure, post-translational modifications, and glycosylation sites of the monoclonal antibody. So during this MAM development phase, we've eventually identified and archived the mass to charges, or the signals that we'll see for each of the individual peptides that are generated from our digest. We can now move into the implementation phase, which is unique to multi-attribute method, where we're actually monitoring just the mass to charge, or the first signals of those peptides. So we're no longer doing collision. This allows for a more robust and accurate quantification of the individual peptides. And you can see then the reference sample versus a test sample, which in multi-attribute method we run back to back. And what this allows us to do is to both check the reference sample to say, yes, indeed, I am getting the relative abundance of each of those individual peptides that I expect, telling me that my system is working appropriately, or my system suitability sample. And then I run my unknown test sample. So I can look at this test sample to do a couple things. So one, I can definitely look at each of those individual signals, ensure that on a mass to charge basis, we're getting the same relative abundance of certain attributes that I want to monitor. But also I can do new and change peak detection to directly compare the test sample to the reference sample without any preconceived notion of what the identities of those peaks are. So I'll talk about that in a little bit more detail in the next couple slides. So this is a little bit of a different representation of mass spectrometry. This is using some software that we've been utilizing in the past couple of years to look at analysis of these peptides. So on the right-hand side, you can actually see that we've sort of moved this chromatogram that we showed on the previous slide to be the y-axis. And now on the x-axis, we're showing the mass to charge of the individual peptide ions. So what you can imagine is each of these individual signals on the right-hand side. Let's look at the tallest peak, for example, on the right-hand side. And then as you move from right to left, you'll see sort of these clusters of individual peaks, right, that are of these different colors. So each of those clusters is actually an individual peptide of a different charge state potentially, or different isotopes of that same peptide. So if we actually circle around one of these groups of ions and we were to fragment that group of ions, you would now see this new box pop up that is actually the fragmentation spectrum of that peptide. So you can see labeled peptide, this is actually that parent ion, and then you see these little labels that say Y and B. These are actually fragment ions where individual amino acids are essentially falling apart from that peptide. And again, this is sort of like a puzzle that we can piece back together to actually get the identity of this peptide. In this case, we have the GPS VF PLAP SSK peptide, which is again a short peptide from the NISTmAb material. It's safe to say that each of these individual spots is representative of a signal in the mass spectrum that we can then identify and associate with a given peptide. And once we've essentially identified these peptides, we can put bounding boxes from where that signal will appear, and we can continuously quantify that peptide based on its mass to charge instead of a UV signal, which is far less selective than all of this mass information that we're able to glean by using the mass spectrometer. So zooming a little bit closer in on one of those individual peaks, this is actually a peptide that we call the penny peptide. As you can see in the upper right-hand corner of this spectrum, the end residue spelled penny K, hence the name. But this is an important peptide because you'll see the ends here are the asparagine residues, and each of these asparagine residues can deamidate. So this is that post-translational modification that I mentioned earlier that has the NH2 group going to an OH group. So what we see moving into this figure, the unmodified arrow points to the parent peptide. But when you have that NH2 to OH, you actually gain 1 Dalton in mass. So now if you look at the D329, for example, you can actually see that that signal has shifted a half of a Dalton forward. This is because we're looking at a doubly charged peptide in this particular case. What we can actually do is do an extracted ion chromatogram of that section, and you can actually see the relative abundance of the various peaks. So these are the iso-D, or the isoaspartic acid eluting before the unmodified peak, the unmodified, and then various combinations of deamidation of individual residues, combined residues in the double deamidation, for example. So we can now associate these relative abundances on a very mass-specific level. We can actually quantify the isoaspartic acid, the unmodified, in each of the individual deamidation events. And we can do that based on mass as opposed to just a signal associated with the UV. So we can actually get much higher selectivity for our quantification to, again, monitor individual attributes of this peptide much more selectively than we could with UV analysis. So then we can do new and change peak detection that I mentioned before. So once we have those bounding boxes in the previous slide, we can actually quantify, again, on routine samples and test samples, how much of the material has actually deamidated and in which fashion. But we don't want to use, we're then having bounding boxes, for example, in the reference slide here. We're not actually using the entirety of this space, right? There's a lot of white space where we don't have any signals. So the question then comes up, if we're not monitoring these individual bounding boxes, what happens if something we're not expecting pops up at a different mass to charge? So we have a process-related impurity or a product-related impurity. In this case, we do a direct comparison using software that essentially subtracts the test sample from the reference sample, and we can have it look without any preconceived notion of the sequence or the identity of that protein and say in your reference versus your test sample, has a signal actually changed? So this is basically asking, are there any new peaks, or did any of the peaks that are in the bounding boxes changed that we may not be selectively monitoring in our report? So this really is an agnostic new or change peak detection that allows you to identify differences in the sample that, if they are actually observed, you can then go in with an additional experiment to elucidate the identity of those changes in more detail. So as you can see, this is an extremely complicated method. There's a lot of data processing, a lot of data acquisition that goes into proper performance of the multi-attribute method. So what we wanted to do was really evaluate how well this assay is being performed across industry, but also identify areas that we can work together to improve the method to really allow implementation in both the product development or process development side, and hopefully eventually into the quality control environment. So with this, we worked with Rich Rogers, now with Bristol Myers Squibb, to organize a combined multi-attribute method. So Trina Muschauer is a colleague of mine at NIST that's been leading this study from the NIST side. And then again, Richard on the BMS side has also been helping, along with the MAM consortium, which we recruited approximately 30 members throughout industry to essentially run the same multi-attribute method, come back together, discuss their results, and understand what our current capabilities are for performing this analytical method. So the scope of the study, again, we really wanted to try to harmonize as many aspects of this to really kind of hone in on the data acquisition component. So in the design, we actually did the digestion at NIST to ensure that there wasn't lab-to-lab irreproducibility there. So every lab was actually sent pre-digested samples, shown in the upper left-hand corner. We actually defined the column and gradient in the blue boxes. So each analytical lab used the exact same UHPLC column and performed the same gradient. What they were allowed to do was choose their own equipment. So they're able to use their own UHPLC, their own high-resolution mass spectrometer. And you can see on the right-hand side that we actually got a pretty good sampling of different types of mass spectrometers. We had QTOFs, linear ion trap Orbitraps, and quadrupole-based Orbitraps. So we tried to, again, limit the variability as much as we could control and leave it to just the acquisition and data processing to understand how this assay was being performed. And in order to do that, we supplied the users a number of samples. So we supplied them a system suitability sample, which was basically just a mixture of individual peptides that are synthetic, that we knew the individual identity of those peptides. We also supplied them a reference sample. So this is essentially just the digested NISTmAb reference material. And then we sent them a couple of these challenge samples to actually look at their new peak detection capabilities. So there were 3 individual samples. one that was a spike sample that had the 15 peptides from the Pierce mix spiked in. We also did a stressed material where the NISTmAb was stressed with pH to induce high levels of deamidation. And then we gave them an unknown sample, which was actually just the reference sample, but labeled as an unknown to test for the potential for false positives. So I won't be able to go through the entirety of this study today. I can give you a very brief synopsis of the results and point you to the paper that we've submitted that should be available in a few months. Looking at the bottom right-hand corner here, we're actually seeing the results from each of the individual participants. So each shape on the x-axis represents an individual pharma lab that produced their analysis. And what we can see in the new peak detection of this spikes sample is that almost every lab was able to identify all 15 peptides, but some of the labs identified additional peptides and some didn't. So some labs found things like modified spike peptides and spike peptide impurities. Some of these we actually did know are present from the synthetic peptide synthesis, but they're very, very low abundance. So again, it really does depend on the thresholds that you're setting of whether or not you detect those peptides. In addition, there were some NISTmAb peptides that were detected as either new or changed. So again, this shouldn't have happened in the individual laboratories, but there was an issue with column carryover, incomplete washing of some of the column components or the autosampler components, et cetera, that we were able to identify. And then there were some unknown M over Zs. Some of these were due to water contamination, system contamination, previous samples, et cetera. So while this figure shows that there was some inconsistencies between the lab, there's a lot of really, really good news here. And most everyone was able to identify all 15 peptides. People were actually setting their new peak detection criteria within the current metrics of how instrumentation was performing. And this was actually based on our system suitability performance across the individual labs. And most importantly, those that were deemed false positives or false negatives could actually be explained. So in the paper, we'll actually go through some of those individual nuances where we were able to dig deeper into the raw data and find some of those false negatives and false positives, and then some recommendations for how to go about watching out for these potential pitfalls that one could run into. So really, the future perspective of MAM, I think, is a very bright potential for an analytical assay. There will definitely be some need for additional validation criteria, very good system suitability controls, but we really do believe that this analytical method has a bright future, and we'll start to see a more broad implementation of this throughout the drug development lifecycle. So sort of a conclusion on NIST's kind of current and future state. Obviously, we've done a lot of work with monoclonal antibodies over the past 8 or 9 years. Glycosylation analysis, mass spectral libraries, you name it, there's been a lot of measurements done to support the biopharmaceutical, specifically monoclonal antibody field, going from reference materials and now all the way up into the process analytics in the upper right-hand side. And with that, we'd like to continue to look at other modalities. We've actually started programs in both CAR T therapies, encapsulated gene therapy, mimetic products, and we're also getting into the RNA and DNA-based vaccines. So we really do have a vision as a national resource that can provide, again, the materials and the science and technology development to support these emerging modalities and also the modalities that have become commonplace and absolutely necessary for a solid healthcare infrastructure. So as a global summary of the NISTmAb, again, we really focused on material properties in releasing this reference material. There's a very large structural knowledge base available from NIST, as well as 20+ publications from stakeholders that have been put into the literature since the material has been released. It's free of intellectual property, so again, anything collected on this material can in fact be published. It's completely open innovation for de-risking analytical technology, and it is a perfect common framework for harmonization and really understanding cross-industry and global trends in analytical measurements. So just as a reiteration as well, we have our program far beyond MABs. We have standards for characterization that we're developing for a variety of different therapeutic modalities. And really, we're working on a lot of advanced analytical characterization standards, et cetera, that can really be used in the future, I think, to characterize these products. And then I'll mention last but not least that industry challenges really do drive the science, technology, and reference materials that we develop. So there is a robust pandemic response as well with the current situation with COVID-19 of NIST, looking at measurement infrastructure standards and technology development that are needed to support that field. So with that, I'd like to thank a very large number of folks that have contributed to this project over the past year. The data I've presented today is probably 300 plus scientists throughout industry that have contributed in some way, shape, or form. But specifically, I'd like to thank the team that is currently working on some of the mass spectrometry and LC characterization, the Biopharma IQ's team, the NISTmAb team that did a wide variety of analytical characterization on the NISTmAb material, and then specifically call out our new peak detection round robin organizers, Rich Rogers of Just Now BMS, and Trina Muschauer of NIST, who has been really leading that effort. So with that, I'm very thankful for the opportunity, and I'll look forward to receiving any questions or comments that anyone may have. Thank you very much.